The title of this presentation is Constructing De Bruijn Graphs from FASTA Input. To begin with a presentation overview, we'll be introducing a GitHub repository, which allows us to automate the process of constructing GFA files, which is a particular type of file format that's related to a FASTA file format, which is used by the genomics and genetics and related communities for storing different sequencing formats, and how this GFA file format, which is used to construct a De Bruijn graph, can be viewed in Bandage. And we'll be displaying the output contents in the command line that are produced from running the script on a sample FASTA file. And this will allow us to provide performance metrics, i.e. the number of sequences produced for constructing the Bruijn graph. And we'll conclude by establishing a connection with Bandage, which is a bioinformatics application that I described in another presentation on the channel that we can use for viewing the contents of the GFA file. So first, to begin, we'll be describing and making use of this GitHub repository for making GFA files. And the GFA file is related to the contents of a De Bruijn graph, which is allowing us to view the overlap between different genetic sequences that we're inputting into the algorithm from a FASTA file. And it's made available to us by this GitHub repository by these two developers, Paul and Bruijn. And first, we're going to be describing the output from the GitHub script that we're going to be using and running the script from. And then we're going to demonstrate how this output that's produced from the script for instantiating and creating the contents of the GFA output file, it's related to the FASTA input that we're using, as well as the individual contents and characteristics of each one of the sequences, which are in the FASTA file that we're using as input. So what we have to do to make the GFA file is that there have to be countably many sequences that are produced for each one of the vertices of the graph from the De Bruijn graph, which is stored in the GFA file format. For one, in the command line, first, there are some S items, some items which are indexed by S, and this is the index of the first sequence, which is just given by some sequence, a DNA sequence of ACTs and Gs, which is output by the algorithm in the command line, which is part of the contents of the GFA output. And we can see that on the slide here, I've demonstrated from the command line what the script from the GitHub repository for producing the GFA output files produces for the first 8 S sequences. And for this FASTA file that was used as input, approximately 370 sequences were produced by the GFA file output. And we can see that as the algorithm continues making these sequences that are going to be stored for the De Bruijn graph, which are passed along to the GFA file output, the sequences themselves get shorter. So they're all DNA sequences because they're still all consisting of ACTs and Gs, but they actually they represent a shorter and less complex assembly, which is representing less complicated components that are used for the De Bruijn graph. And besides the S elements that are created in the command line for the GFA output, the script from the GitHub repository that we've been referencing from the two developers that we're using with our FASTA file, it's also iterating over each vertex of the graph, and it creates some other elements, namely the L elements. And these L elements are a little bit different because they're not consisting of the GFA or the actual sequences that are passed from the GFA file output, but rather they're consisting of characteristics of the length of the k-mers, as well as some other qualities which are used to describe how the De Bruijn graph and its structure can be viewed in the Bandage application, which we're going to refer to and allude to at the end of the presentation. But what about the FASTA file? So we also, in comparison to some other open source and bioinformatic pipelines, which describe FASTA files, but may not provide an example or actual contents of the FASTA file, I wanted to describe and provide an example of the exact sequences that are given in the FASTA file that I use as input into the Python script, which are used to produce the contents of the GFA file format, and to describe how this relates to the TXT file from the FASTA input that we're using. So first of all, we can see that the FASTA file is given by a collection of DNA sequences, and the DNA sequences appear at different points in the FASTA input, which is the TXT file. But the difference is that they have a unique identifier or a unique barcode at the beginning of every one of the at the beginning of every one of the DNA sequences, which is given by an ampersand and a unique barcode, which is describing different types of characters which appear throughout the DNA sequence, as well as relating to the overall length of the DNA sequence. So there can be DNA sequences that could be hundreds or thousands of base pairs long. In this case, to simplify the process of compiling the script to obtain the GFA output, I used some simpler, shorter types of DNA sequences just to see approximately what would the runtime be and to be able to achieve a reasonable runtime for the algorithms for constructing the GFA output from these DNA sequences. And we can even see that more near the middle of this slide, there are some DNA sequences that are a line long in comparison to, or as opposed to, other DNA sequences that can be a couple of lines long. So what the algorithm has to do, as we're going to highlight and describe in previous slides from the script in the GitHub repository, is that it's looking at a fixed k-mer length, and it's looking at overlapping reads of the genome and trying to construct and help us visualize and more clearly understand structure of the whole genome from shorter term reads and overlapping hundreds or thousands or hundreds of thousands of these shorter term reads to obtain a more clear resolution and a more clear a whole sequencing of the entire genome from sequencing along these fragmented segments. And the end of the FASTA file looks like this, in which we can see that we also have, as we had mentioned previously, the unique barcode identifier at the beginning of each one of the sequences, which begins with an ampersand, and it has different types of characteristics, namely the types of DNA base pairs in the order sequence in which these base pairs occur, as well as how long the sequence is itself. So this is just giving an idea of different types of characteristics in the specific formatting of any one of the DNA sequences which appear in the TXT file from the FASTA file that we're using as input to make these test GFA files that we're going to be able to view and look at more closely in another bioinformatics application. So the first few methods of the script search over the base pairs of each input sequence from the FASTA file. So it's taking the base pairs of the sequence, and depending upon how long we set the k-mers to be, whether it be 30, 31 is a typical value that many people use when looking for k-mers. There can also be shorter values for the k-mers because you can pass any value of the k-mer to the script. But in this case, I did 31. And then it's looking at just basically mod 31 of the length of each DNA sequence, how many characters there are, and being able to look at each one of the DNA sequences in compartments of 31 base pairs long, namely 31 base pairs where each one of the base pairs can be an A, C, T, or a G. And we're looking at constructing k-mers, and we're looking at being able to determine how the sequence would change from some fixed information that we have in the sequence as we're looking at what entries or base pairs are in the sequence from whatever values of k-mers we're looking at. And we can also look at other methods later in the script to give us more of a clear idea about what this GFA file output is and how it relates to the de Bruijn graph and how we can look at it in this bioinformatics pipeline in applications that we've been discussing, in which we want to look and manipulate sequences for a fixed k-mer length and to construct contigs. And contigs are used in many types of bioinformatic pipelines. And even another presentation that I've given on the channel, we talk about transcriptome assembly or genome assembly, and contigs are assembled and used in different types of bioinformatic pipelines for being able to form well-guided predictions about genetic content of different sequences and whether there be any conserved sequences, what would be the total length of the sequence, what would the sequence begin with, or what would the sequence even end with, and related types of questions that we can think about. So the thing is that we can look to build different types of sequences which are going to populate different entries and parts of the de Bruijn graph by the build function, which the developers from the GitHub repository include in their script. And then basically they also have another method which allows us to convert the config to a string. And then we can also have, they also have a method for us that allows us to retrieve the config after it's constructed. And as we have been mentioning, the GFA files can be viewed and manipulated in Bandage. And I talked about Bandage in another presentation on the channel, which isn't too long. It's a shorter presentation in which I drew the viewer's attention to visualizing de novo assembly graphs with Bandage, in which it's basically a way to assess different types of sequencing quality from graph structure. And the Bandage application can also be used to look at sequences, the graphical structure of sequences directly from FASTA files, or can also be used to look at the sequence structure from GFA files from the de Bruijn graph. So it's a pretty robust bioinformatics application, and the type of output that we've obtained from the GFA file from the command line that I've described for you in this presentation, it can also be viewed and looked at in this Bandage application, which is another bioinformatics pipeline. So it's very much an application or it's very much a demonstration of how one can make use of and blend different bioinformatic pipelines together for performing various analyses depending upon the application that someone's interested in. And a conclusion is that we introduced a GitHub repository due to the two developers that I named at the beginning of the presentation for producing GFA files, which can be viewed with Bandage. And we described the contents of the script that were produced in the command line. But besides these contents, which are used for the GFA file format, we also provided examples of sequences from the FASTA file that we used. And we used simpler sequences just to make this script be able to compile within a reasonable runtime from the beginning to the end, which are used to construct the de Bruijn graph in the GFA format, as well as we provided and drew the viewer's attention to one potential bioinformatics pipeline or application, Bandage, which can be used to view the contents of the GFA file. And thanks for watching.